today's topic is on the gram staining. Okay, so you will be doing the gram staining yourself. This is gram staining. Okay, can anyone tell me what is this color? Purple. So I have made this purposefully so that you guys remember, in case of gram staining, never use the blue color. Okay, gram positive will always be purple or violet. Okay, in case of the stains which stain the smear into blue, will have the blue in their name. For example, what are the stains you know other than gram staining? Methylene blue. Methylene blue has methylene blue in it. Then the other one you read it in mycology. No, crystal violet. No, crystal violet is used in gram staining, and in the name there is violet, so the color will always be violet. The other one that you read in mycology was LPCB, lactophenol cotton blue. So only those which have the name, in which the names already contain the word blue, will only be blue. In case of gram staining, gram positive will always be purple or violet. Okay. So you already know about the simple staining. Okay, so I have already given it the color blue because we use the methylene blue there. Okay, negative staining, you guys know about this. Can you tell me an example of an organism that we saw it under negative staining? Cryptococcus. What was the negative stain called as? India ink. The other one? Nigrosin. Very good. Then we have the differential stains. Can anyone tell me what is the meaning of differential staining? Two colors. It divides the bacteria into two different groups. Okay, two or more different groups. So in that we have gram staining that we'll be reading today, and the Zn staining. Okay, and finally the special staining. Do you guys remember the special staining that we told you guys? Albert stain. Very good, exactly. And can anyone tell me what was the organism that we saw there? Corynebacterium diphtheria. Okay, and the pattern was Chinese letter pattern. Okay, and or the drumstick pattern. Okay. So we'll be learning the procedure, theory and examples. So the theory behind the gram staining, broadly we can classify into two schools of thought. One is the pH theory, the other one is the cell wall theory. Okay, the pH theory is the old one. You just need to know about the name. Okay, in case of cell wall theory, we all know that gram positive have a thick peptidoglycan layer. Okay, so due to that thick peptidoglycan layer, they can hold on to the iodine crystal violet complex more easily. Okay, but in case of gram negative, they have a thin peptidoglycan layer, so they can't hold on to the color that much easily. Okay, so that much only the principle that you guys need to know. You just need to know about the name of the other theory, that is a pH theory, and the theory that we currently follow. Okay, this was the cell wall theory. Okay, you guys get it now? Gram positive has thick peptidoglycan layer, so it holds on to the color more easily than the gram negative ones. Okay, so can anyone tell me what is the difference between a cell wall of a gram positive and a gram negative? Any one point?
In positive we have peptidoglycan,
thick peptidoglycan, and the gram negative is thin. Okay, anything that gram positive has but gram negative does not? Anything with the, yes, Kushmita? Teichoic acid. Okay, gram positive has teichoic acid while gram negative does not have. So that's for gram positive. Anything that gram negative has but gram positive does not? Anyone? LPS, lipopolysaccharide, is present in case of gram negative but absent in case of gram positive. Okay, anything else? Porins and periplasmic space. Okay, periplasmic space and porins are present in case of gram negative, but these are absent in case of gram positive. Okay, so gram staining developed by the name only there, na? Hans Christian Gram. It's not village. Okay, it's based on his name. Okay, so purpose is to differentiate into two broad groups. We already know, because differential staining, it's a differential stain. So there are four parts in case of gram staining. First is the primary stain. Primary stain, we mainly use crystal violet, methyl violet and gentian violet. Okay, if you are answering very good now, they will ask you some honors level question. The basic question is, what is the primary stain? What are the primary stains? So you can definitely answer this. So the honors level question will be, can anyone tell me the difference between, what is the difference between crystal violet, methyl violet and gentian violet? So remember, crystal violet and gentian violet are both the same thing, but methyl violet is actually the whole group of para-rosalinidine dye is called as methyl violet. Under that, we have many branches, okay, based on the number of molecules, hexapararosolanin, pentapararosolanin, okay, octapararosolanin. So, crystal violet and gential violet are the same names, but methyl violet is a family name, okay. Consider that methyl violet is like the surname, okay, everyone has the surname in your family, same, but crystal violet and gential violet are your actual names, okay. Then we have the grams iodine, which is a mordant. Mordant means to fix, okay, to stick or something like that, okay. So grams iodine act as a mordant and it forms a iodine crystal violet complex. That complex gets trapped in the thick peptidoglycan layer of the gram positive bacteria, which ultimately gives the color purple, okay. So grams iodine. They will also ask you what is the difference between grams iodine and another iodine that you guys used in parasitology. Do you guys remember what is the iodine? In a procedure used in parasitology. You guys have also written the differences between these two procedures, saline mount and iodine mount. So what is the difference between the grams iodine here and the iodine used in case of iodine mount? Grams iodine is a much more higher concentration of iodine, has a much higher concentration of iodine compared to the iodine that we used in iodine mount, used in parasitology, okay? Can anyone tell me the difference, what is the main advantage of using iodine mount over saline mount? Listen to the question first. I ask you what is the difference, main advantage of using iodine mount over saline mount? Nuclear details of cysts and eggs and larvae of helminths can be more easily visualized. Very good. Final, the third step is decolorizer, okay? Remember, it is the most important step, okay? We'll learn about it, okay? Decolorizer is the most important step. We either use acetone or use alcohol, okay? And finally is the counterstain, okay? We use safranin or we use Carbol fusion. We used basic fusion in which staining? Simple staining, very good. So the procedure, the first step is already done for you, the smear making. In case of smear making, just know the steps. We take a clean, non-greasy slide, okay? And then we put a drop of saline water over it and we take a colony out using a inoculation loop, okay, very good. Inoculation loop, and we emulsify on the saline drop, okay? We move it in a circular or an oval shape from center towards the outwards, okay? So our talented lab technician has already made the smears for you, okay? So finally, you guys also know about this. We have read this in the morphology chapter now. The different shape gram-positive cocci, gram-negative cocci, bacilli and gram-positive bacilli. So you guys need to also know about the examples. Some of the examples are very common. Na? Staphylococcus aureus, Streptococcus pyogenes, these are very important. In case of gram-positive bacilli, we have the Bacillus anthracis, Corynebacterium diphtheria, Albert-Stein. Gram-negative, remember these three are also very important. Okay, Neisseria meningitidis, Neisseria gonorrhea. Okay, and finally, in case of gram-negative bacilli, remember to always say Escherichia coli, because there is another coli in parasitology. That is also E. coli. That is Entamoeba coli. So if you tell E. coli, na, the external or internal who will be taking your viva, he will catch you there and tell you, so are you talking about the parasitology or bacteriology? So always tell Escherichia coli. Okay, Salmonella typhi, Klebsiella pneumonia, and Proteus vulgaris. A lot of you are saying just simple Proteus. Okay, you have to either say Proteus mirabilis or Proteus vulgaris also. And finally, you just need to know the names of these modifications. Okay, Kopelov and Biermann's modification. These are some small changes in the gram staining. The main theory remains the same, but we either change the decolorizer or the staining or the counter staining one. Okay. We have also got the Wiggert's modification, we got the Jensen's modification, where you use absolute alcohol instead of a acetone, okay? We have also the Preston and Models modification, okay? Okay, so this is a gram staining chart. The first step is the smear is stained with a para-rosolanin dye. Para-rosolanin dye are, all these are para-rosolanin dye, okay? Methyl violet, gentian violet, crystal violet. For 1 minute. First step is primary stain, okay? Primary stain, we use either methyl violet, gentian violet, crystal violet, and we use it for 1 minute, okay? After that, we rinse it under the water. We'll show you guys how to rinse it properly under the water, okay? The second step is the mordant, okay? So after we have washed away the primary stain, we'll flood the smear using grams iodine for again 1 minute. Remember the times, very important, okay? Iodine serves as a mordant, forming larger and more stable complexes within the bacterial cells, okay? And finally, again, we'll be rinsing it under the water. After that comes the most crucial step of gram staining, okay? Remember this. Decolorization is the most crucial step of gram staining, okay? We use either ethyl alcohol or we use acetone. In case of acetone, if we use acetone, we'll just keep it for 2 to 3 seconds, okay? But in case of ethyl alcohol, we'll be keeping it for 20 to 30 seconds. In our lab, we'll be using the acetone. So remember, we use it for only 2 to 3 seconds. And the final step is the counter staining, okay? So counter staining can be Carbol, Fuchsin, Safranin, and we'll put it for 30 seconds. So repeat after me. The primary stain, we use crystal violet for 1 minute. In case of Mordant, we use gram iodine for 1 minute. In case of decolorization using acetone, we use it for 2 to 3 seconds. In case of ethyl alcohol, 20 to 30 seconds, okay? And finally, the counter staining, we use Carbol, Fuchsin or Safranin for 30 seconds. So remember, in case of gram positive, okay? In case of gram positive, as they have a thick peptidoglycan layer, they will hold on to the color more easily and will not get washed away when we are decolorizing, okay? So the complexes will already have that color and the safranin can't enter. So in case of gram positive, we will always be having a purple or violet color. But in case of gram negative, in the decolorization step, as they have a thin peptidoglycan layer, they lose the color very easily, okay? So after that, as they have lost their color, they are colorless, so they take up the counter stain pretty easily. So gram negative will always show a pink color, okay? And finally, these are the photos, okay? This is how it's supposed to look, okay? Can you see that these are the pink and this is the violet one, okay? So that's it. Now we'll go and do the staining ourselves, okay? Flood the slide with water directly, okay? You're gonna hold it like this at an angle, not like this, okay? Otherwise, the whole smear and the color will come to you. The biggest flex in case of doing a gram stain is to show that your hands are not dirty. That is the biggest flex, okay? Because everyone's hands will be dirty and your hands will be very clean. So remember this, hold it like this and let the water dribble down through your index finger onto the slide, okay? Or alternatively, you can also pour the water at the end of the slide, okay? But this is gonna be a bit difficult for you. So you're gonna do it like this, okay? You will hold it at an angle, okay? So let's start the first step. First step is the primary stain, okay? Remember, the time starts from the moment you drop the first drop, okay? Sometimes you have to squeeze the bottle hard because the liquid is the whole circular area you can flood, okay? So now the other one can start also, if you are keeping a track of the time. Everyone has to keep track of their own time, okay? Okay, the Mordant is the second step, so you're gonna pour the Mordant over there and keep it there for 1 minute, okay? You are keeping track of your time now. You are also keeping track of your time. So, what is the second step?
Mordant.
Mordant, okay? So take the Mordant. Is your time over? So you need to wash now. So you need to wash. You're gonna hold it like this, you see? You take it from here, okay? And you hold it like this, okay? Then you can wash. Hold it and wash. Okay. I think that's enough. Okay. So pass on to the Mordant him. So remember, you're gonna hold it like this, okay? See? Why are you guys taking the long way route, then holding here, okay? You're gonna just hold it from here. You're gonna take it here, okay? Open the tap a little bit, okay? See? That's enough. That's enough, okay? Mordant for 1 minute, okay? Take the Mordant and keep it for 1 minute. Is your 1 minute over? Place the Mordant and keep it for 1 minute, okay? Do? Mordant, take the Mordant. Yours one is done now. After that, you will be doing the Mordant step, okay? Pour the Mordant over there. एकदम से ऊपर कर दे, फिर स्क्वीज कर, हल्के से स्क्वीज कर, हाँ. चलो, टाइम देख रहा है ना तू? ठीक है? टाइम देख एक बार, ठीक है? अभी तुम्हारा पार्ट है. It at a very low speed, okay? Okay. I think that's enough. You're gonna pour the decolorizer and you're gonna wash it Immediately. You're not going to wait for some time like 2, 1, 2, 3. No. You're going to pour 2, 3 drops and you're going to wash it directly because 2, 3 seconds is a very short time. Okay? You wait, okay? Wait. I'm going to come. Wash it directly now. Go. Wash it. Very good. I think that's enough. Now there is a counterstaining. Counterstaining using safranin for 30 seconds. Okay. Start. Is your time over? Okay. You're gonna pour the decolorizer. Bas. You can wash directly using the same technique. Okay. Okay. I think that's enough. Now we're gonna do the counterstaining using safranin for
30 seconds.
Okay. Do it. Have you placed the Mordant?
No, sir. Yes, sir.
Okay, Mordant. Now you're gonna do the decolorizer. You have to wash it away now first. Have you washed it? So wash it away. Okay. Is your time over? So you're gonna wash it. Just I told you. Okay. Now your step is the decolorization.
Yes, sir.
So you're gonna take this one.
I have to wash it instantly.
You're gonna put 2, 3 drops. You're gonna wash it instantly. Okay. I think that's enough. You're gonna wash it immediately. Very good. Okay. Just hold it a little bit higher. Otherwise, okay. I think that's enough. Okay. Are you guys doing the decolorization now? Okay. 30 seconds gone now. I think your one is washed away a little bit. Okay, we're gonna wait. We'll wait for the excess water to dribble down. Safranin, where is the saffron? This is the saffron. Okay, counterstaining for 30 seconds. Okay, let's go. Don't wash away too much, otherwise the whole smear is gonna get washed away. I think that's enough. Now what do? You've done the counterstaining now? You're just gonna place it here to let the excess water dribble down. Okay. अभी तुमने decolorizer यूज किया? कितना टाइम के लिए किया? डालने के बाद immediately धो दिया ना? ठीक है. अभी तुम saffranin डालो. ठीक है? हो गया. Don't wash too much, otherwise everything is gonna get washed away. Now we're gonna place it in that crevice, okay? So that the excess water dribbles down. You're gonna do the same, okay? Tilt it a little bit and place it just between the faucet and that basin, okay? You don't need to do this, no? You can just place it here. Just you wanna wait for some time after the excess water has been. You have done the counterstaining, okay? So what you're gonna do is you're gonna put it in this crevice between the faucet and the basin, okay? So the excess water dribbles down. After that, you're gonna do after the excess water has dribbled down, you're gonna do the air drying, okay? So you guys know how to do the oil immersion objective, how to focus. So you're gonna do it yourself today, okay?